Welcome everyone to the fifth in a series of joint research symposium between Rutgers University and Tel Aviv University. My name is Vince Miraglia from Rutgers, and we're going to have two speakers today on the topic of advances in gene therapy. Just a quick overview of the program today and some instructions on how to ask questions during the Q&A session. We'll first hear today from the professor of Department of Human and Molecular Genetics at Tel Aviv University, Dr. Karen Avraham, who'll be talking about gene therapy for deafness. And afterwards, we'll hear from Maral Mouradian, professor of neurology at Robert Wood Johnson, on gene therapy for treatment of complications of Parkinson's disease. And then we'll engage in a Q&A session for both of our speakers. If you have a question anytime during the course of this event, please use the Q&A button you can find on the bottom of the Zoom presentation, and you can start writing those questions as they occur to you, and I'll work to collate those questions and start to ask them after both presentations have been completed. We're going to be recording this session, so you can tell colleagues who missed it that they can go to a website that I'll announce at the end to be able to see this seminar series as well as the other four that came before it. Right now, it'll be my pleasure to introduce you to Dan Pierre of the Tel Aviv University, who's going to introduce our first speaker. Dan?
Thank you, Vince. So I'm very happy to be here again. And our first speaker is Karen Avraham. And Karen is a human geneticist. She was born in Canada and moved to the US at a young age. She holds the Dr. Sarah and Felix Dermott Chair for Research of Hearing Disorders. And she's also the Vice Dean for Research at the Sackler Faculty of Medicine. So she has done pioneering work in the field of gene therapy to deafness, and she's also a member or a council member of the European Molecular Biology Organization, the EMBO, since 2017. And she's done a lot of other very interesting work with long non-coding RNAs and methylation in auditory system. She's been awarded the Sir Bernard Katz Prize from the Alexander von Humboldt's Foundation in Germany, and the Bruno Memorial Prize from the Rothschild Foundation. Also the Teva Prize for groundbreaking research in the field of rare diseases from Teva Pharmaceuticals, and the Teva Founders Prize on breakthrough. I'm looking forward to hear Dr. Avraham's work. Thank you.
Thanks, Danny. And I have to give a personal shout out because I actually spent many of my formative years in New Jersey. I lived in Parsippany and Elmwood Park and North Caldwell. And so I have wonderful memories from many years of living there till I went to Washington University in St. Louis. And then I came to Israel and did my PhD at the Weizmann Institute, did my postdoc at the National Cancer Institute, and I've now been at Tel Aviv University for 25 years, almost going on 26. So it's wonderful to be here and to be part of this group, and in some ways to come back home for just a little bit. So let me share my screen with you, and I'm going to give you a brief presentation on the work that we've been doing in our lab over the past couple of years. And I know Dan said he used the word Karen, but I want to emphasize that the work that I'm talking about today is incredible team effort and primarily done by one MD PhD student I'll tell you about in a moment. Okay, so I hope I'm on share screen and you can see my slides now. And so again, this is really exciting for me to be here and almost be back to New Jersey. So why hearing loss? Why is this an issue that we've been working on for, actually I've been working on it already for 30 years. It's because 500 million people worldwide have a disabling hearing loss. This is an enormous part of the world. I don't probably have to go into details about the difficulties in communication, in rehabilitation, in development, the economic and cost, the burdens to society. And while there is a group of individuals who believe that deafness is a different form of communication, the better part of those who have a hearing loss are very much impacted by it. To the extent that the Lancet has put together a commission on hearing loss in the last year, I'm very proud to be one of the advisors there. And our vision is a world in which hearing loss is no longer a barrier to human communication and fulfillment. And it's also very much part of the Sustainable Development Goals that's put out by the United Nations. And even more so, it seems that about 1 in 4 people are projected to have hearing problems by 2050. So it's an enormous problem. And most of what we've been doing in the last 20, 25 years or so has been concentrating on genetic hearing loss. This is over 50% of hearing loss. And even that might be primarily environmental due to autotoxic drugs or elements that can continue upon aging. A better part of those will still have a genetic susceptibility. What you see here are all the human chromosomes, and randomly distributed throughout them are the different genes that are involved in hearing loss. Now, I can give an entire talk about just one of these genes, but I'm going to concentrate primarily on the one that we've done gene therapy on. But just to really give also acknowledgement that much of what we were able to do in the last years was because of the enormous development in the Human Genome Project. And that project now just celebrated 20 years. And so we've become very aware of the work that's been done and just how much this has given us to advance, not only in identifying the genes, but also discovering the mechanisms that are involved in all of these different pathologies. So the inner ear is really a remarkable structure. It's made up of both the vestibule, which is responsible for balance, and the cochlea, which is responsible for our hearing. And if you go further and look deeply into the cochlea, which is the part that contains our sensory cells of the inner ear, you can see these really beautifully arrayed 3 rows of outer hair cells and 1 row of inner hair cells. And those together really allow hearing to occur in a really highly structured and organized way. Now, if I'm going to show you what it looks like at the level of a cochlea, you can see what we're looking at here is a mouse cochlea, and you're veering in to these really beautifully arrayed inner and outer hair cells. And I'm going to talk primarily about the outer hair cells in a few minutes. What I also want to define before we go into the details about hearing is the different forms of gene therapy that are going to be used. Now, we're going to hear later from my partner a lot of gene therapy that's being done and work that's being done on Parkinson's disease, but I want to just focus on the 3 major types of gene therapy, gene or cell therapy that can be done. One of them is to use antisense oligonucleotides. The other is one that I'm sure many of you heard about, is CRISPR-Cas9 genome editing. And in fact, Dan Peer, who introduced me a few moments ago, has done some really remarkable work in this area for cancer. And then the third, and that's the method of choice that we used, was adeno-associated viruses. Now, I'm not going to go into details because of time about the others, but I do want to focus on the adeno-associated virus, or what we call AAV. And the reason that it's been so promising is for several reasons that I'll talk about in a couple of minutes. But I do already want to say that there are about 200 clinical trials that are ongoing using AAV. And there is currently AAV gene therapy both for spinal muscular atrophy and for Leber congenital amaurosis. And so this is already being done in the clinic. And what I'm going to talk to you about is the work that we've been able to do in our research lab. Now, there are many considerations for therapy, and we'll hear more about some of the clinical applications later. But first and foremost, it's very important to understand the mechanism. Why, whatever it is that you're looking at, why is it occurring? What's the phenotype? When does the damage occur? What are the genes that are involved that might need to be corrected? What are the targets that might have to be also impacted when you're starting to look downstream at some of the events? Especially if we're talking about CRISPR-Cas9, what are the off-target rates? We want to make sure that we're not mutating other regions that can then also have a deleterious impact. We want to be able to provide therapy that's going to be sustained. We don't want it to just be for 1 month or for 1 year, but we want to have as many indications as possible, just as we want with the new Pfizer and Moderna vaccines, that they're going to be able to last for as long as possible. How are we going to deliver these different forms of therapy, and what's the strategy? So I just want to turn your attention to this very nice image that was taken from a review that came out just a few months ago. And it shows you some of the possibilities for how you can provide gene therapy in the inner ear. And I think it's important to emphasize that there are different areas in which we can target these different hair cells. Now, the story that I'm going to briefly tell you about for gene therapy really started in early, I would say probably about 2009, when a physician that we work with, Professor Moshe Friedman, brought us these two families, and they originally are from Iraq. They're living in Israel for many years. And the type of hearing loss that they have is primarily a high frequency hearing loss. The hearing loss, though, starts very early on. Is inherited in the recessive form, and you can notice that these double lines in the parents because they were actually related in each family. So there was a consanguineous marriage here, and the children, and you can see the number of individuals who inherited this hearing loss. We approached a laboratory in Singapore, and we obtained knockout mice for this gene. The gene is called SYN4. Which actually encodes a protein that's called Nesprin. Now, Nesprin is very important because it's partly responsible for anchoring the cytoskeleton to the nucleus. And so without that, you essentially would lose the ability for your nucleus to be anchored where it should be in the cell. Now, if you look at the bottom here, you can see that the mice shown in red are actually profoundly deaf by the time they get to be 2 months of age. And we believe that primarily this is being caused because the nucleus of the outer hair cell, which on the left side you can see very nicely is at the base of the hair cell, because it loses this Nesprin and you no longer have the anchoring, it now moves to the top of the hair cell. And as a result, these cells aren't functional. And I want to just acknowledge that much of this work in identifying the mutations in this family was done by Dr. Tzipi Brownstein in our laboratory. Well, a couple of years ago, Shachar Taiber, very talented MD PhD student in the lab, came onto the scene and with his sights on doing clinical work in the future, thought that it might be time for us to get into gene therapy. So we joined forces and a really wonderful collaboration with Professor Jeff Holt, who's at Boston Children's Hospital and at Harvard. And we decided to take these mice that have mutations in SYN4 and to try to rescue their hearing. Now, I'm already going to give you the punchline because you can see the very beautiful cover that came out a few months ago in Embo Molecular Medicine, and we did succeed in rescuing the hearing loss. But let me tell you a little bit more detail about how we did this. So first, our approach was, as I mentioned earlier, to use the adeno-associated virus. These viral vectors are linear single-stranded, and they, one of the perhaps advantages or can be a disadvantage if you have a large gene is it only carries up to 4.8 kb, but SYN4 is much smaller than that, so we didn't have any problem with respect to size. These AAVs are relatively non-immunogenic and they remain in the extra chromosomal state, very rare that they undergo integration. So they're really quite safe, and hence the reason they're in so many clinical trials and already being used clinically. So this was the approach that we decided to take. Now, there are a number of questions that we need to answer before we actually embarked on the gene therapy. We needed to first understand what the expression was and make sure that the cells that we were targeting is where SYN4 is expressed. And together in collaboration with Matt Kelly at the NIDCD and looking at a really wonderful resource called GEAR, which comes from the University of Maryland, the Gene Expression Analysis Resource, we're able to determine that SYN4 is relatively restricted to the hair cells. It has higher expression in the outer hair cells, which makes sense given the phenotype that I showed you a few moments ago. And then what we needed to do is actually figure out when was the damage actually occurring? When is the best time for us to be able to introduce the gene so that we can provide the therapy that's very much needed? So what we did is we first analyzed the hair cell survival in the inner ears at different ages. And here you can see what we did at postnatal day 8. We also looked at day 10, day 12, and here you can also see 14. And while at postnatal day 8, the outer hair cells appeared intact, they look really very beautiful, the 3 rows of outer hair cells that we would expect to see. By postnatal day 14, I think it's very clear that you can see that in the mutants that the degeneration was very readily apparent. So we were able to determine the time window in which we should go in and do the rescue. Now, we also wanted to look at the hair cell counts, and we found that there was a very strong correlation, not only with what we were seeing morphologically, but also with respect to the counts. And we also wanted to see whether the cells are still functional. And we used FM143. This is a dye that can enter the hair cells through the sensory transduction channels. It's used as a proxy for functional hair cell sensory transduction. And we found that the SYN4 hair cells can take up this dye at postnatal day 8 in a way that can also the wild type cells, which suggested these SYN4 cells at postnatal day 8, while they have the properties of functional hair cells prior to when they start being damaged. Interestingly, we also looked at the vestibular system where SYN4 is expressed, but these mice don't have any vestibular abnormalities. So our approach was to take a viral, an AAV vector called AAV-PHBP. And the reason that we chose this one is that it previously been shown that it works very well to transduce both inner and outer hair cells at very high rates. So Sackler constructed a vector with AAV and containing SYN4. He first checked to see that the expression of GFP in this AAV is reliable in the hair cells. He could see very clearly, if we look at the organ of Corti injected with AAV GFP, that we have really beautiful transduction in the outer hair cells. And in fact, when he looked at both the inner and outer hair cells, he found he had about 100% transduction rate. So he then cloned the turbo GFP into an AAV2 backbone, downstream of CMV enhancer and promoter, and upstream to poly-A sequence. He packaged the whole construct into these AAV-PHB capsids, which we now call the AAV-GFP. And then he cloned the coding sequence of SYN4 into the backbone, downstream of the CMV enhancer and promoter. He added a flag tag and put the construct into the capsids. So in order to be able to check the transduction efficiency, he also looked very carefully at the inner and outer hair cells, and with the flag tag was able to see that the expression was very nicely in the outer hair cells. When he then looked very carefully at the outer hair cells, we could see that SYN4 rescued the outer hair cells very carefully. And if you look at the postnatal day 14 mice, you could see that the nuclei are at the base of the hair cells. At postnatal day 14 mutants, you can see that these nuclei are already moving up, almost getting to the top of these cells. And if you look at the rescued mice, these SYN4 mice, you can see that once again, these nuclei are very nicely at the base of the cells. And he was able to quantify this with image analysis. And again, you can see very nicely that the nucleus is where it's supposed to be, very similar to what's happening in wild type cells. If you look at just a immunofluorescence showing you the structure of the cells, you can see that they're really quite beautiful and hard to believe that these came from a mouse, that the cells were almost completely degenerated and the mice were deaf. Now, to show you how it manifests itself in hearing, now you can see what we call an ABR, which is a indirect measure for hearing. And if we look at the wild type mice in green, and we look at the mice that are deaf in blue, you can see that if we take the AAV with no gene, just with GFP, just to make sure that there is no damage that's occurring due to the AAV, you can see that that the mice continue to be deaf with the AAV only. As soon as we introduce the SYN4, you can see that very nicely that the hearing is rescued at a significant level. We checked this at 4 weeks, but we also went up to 12 weeks of age and found that this was sustained. I want to emphasize that we also looked at DPOAEs. Now, DPOAEs measure the function of outer hair cells, and since it's primarily the outer hair cells that are damaged here, this was very essential to check, and these also really went almost back to normal, not only at 4 weeks, but up to 12 weeks of age. Now, the treatment really does work from a long-term perspective. If we look at these at these hair cells already at 12 weeks of age. You can see the wild type cells and the very nice form of the 3 rows of outer hair cells. And you look at the rescued, you can see that the hair cells look almost identical, as well as the localization of the nucleus at the base of the hair cell and quantified by image analysis. I want to also emphasize that the inner hair cells of these mice were not damaged very much. That came as a surprise to us because there is expression of SYN4 in the inner hair cells. Though later on, if you look at these mice, there is damage to the inner hair cells as well. So that's what we managed to do, and it's a long road to really finesse all of the details, but in order to be able to rescue the hearing loss in these mice. Now the challenge has become, how are we going to transition this and other work that's being done in humans? So I want to also emphasize that there's other work that's being done worldwide in many labs. There's most recently in vivo base editing has been done to restore sensory transduction and improved auditory function in a mouse model of recessive deafness, mouse model called with a mutation in a gene called TMC1. And about a year and a half ago, and while they're not ongoing anymore, there was a gene therapy trial for hearing loss using A2H1, and it gives us a lot of promise that we can move into clinical trials in the short and the long future. Now, where are we taking this work in our lab is we're working also with not only one of the HMOs, one of the largest HMOs in Israel, where we're looking at all of the individuals there who have a hearing loss, and we've identified thousands of individuals out of the entire biobank who have a hearing loss. We're working to see how many of those have a genetic basis using whole exome sequencing. Those genes that we identify, obviously only a subset of those, we're going to generate CRISPR-Cas9 mutant mice. So they'll be similar in phenotype to what we're seeing in mutants, much the way that we were able to study the mouse knockout for the SYN4 related deafness. And we hope in several of those cases to be developed gene therapies, and we may expand further from AV and use some of the other newer technologies or new technologies that have been used as well. We're also conducting a newborn screening pilot with the Ministry of Health to look at how we can really streamline after the newborn hearing screening that infants fail in the hospitals, to be able to very quickly be able to determine the genetic basis of the hearing loss so that we can work towards rehabilitation as easily and as quickly as possible in these children so that they'll have the rehabilitation that they need. So what are the questions or what are the challenges that we have in moving to humans? And we'll hear more about what's being done in humans in the next talk. But it really requires that the surgery that'll be done will be minimally invasive. We want to be able to target cells along the entire length of the cochlea. We have to be very clear about what the target population is. Are we looking at children if we're talking about a particular gene? Are we talking about those in a later age? Are we talking about age-related hearing loss, which is actually the largest group of those who suffer from a hearing impairment? Are we looking at a type of hearing loss that's genetic, in which case gene therapy will be very useful? Are we looking at an environmental one that we're going to need to figure out what those causes were before that we can deliver a cell and a gene therapy? And of course, it goes without saying, not only for this, but for all types of therapy, we need to be able to establish the safety, the efficacy. So I think that we can be really heartened this year after seeing how quickly we were able to put together work that was done for vaccinations, that we know that we can establish these types of guidelines relatively quickly. And I mentioned it when we were talking about SYN4, about when are we going to do the delivery. And in the case of those mice, the delivery was at postnatal day 0, just the day that they were born. And we're going to try different ages to see how late we can go to be able to still prevent the damage that starts occurring at about postnatal day 10 and postnatal day 12. But we won't be able to do these types of experiments on humans, so we'll have to work out all of these details, not only in mice, but also on non-human primates. We've also been, other than just doing gene therapy and other work in genetics in our lab, we've also been looking at COVID. And we wanted to understand what are some of the boundaries in disseminating information, and we joined forces with Dr. Amiel Jor. Years ago, he did an MD PhD in our lab. He's now a resident at the Galilee Medical Center. And we published our work, and this was really a call for the ministries of health throughout the world to be able to make sure that the websites that they're putting out there are accessible for those who have disabilities. And I'm proud to say that it's had almost over 1 million views to date. We've also looked at the auditory performance in recovered SARS-2 COVID patients. And I'm glad to say that it appears from all of the work that's been done, and he's already looked at hundreds of patients, even though the publication had a handful of patients, that it doesn't seem that SARS-2 actually impacts hearing. So I think that that's good news for those recovering patients. And finally, before I turn it over to Parkinson's disease, I actually wanted to let everybody know that's hearing today that we do have a Center for Parkinson's Disease Research that's very active. It's the Aviv and Family Center for the Prevention and Treatment of Parkinson's Disease. And with your permission, I'm going to show you a very short video, and I think that this will, set the tone very nicely for our next speaker, who we're going to hear from about Parkinson's and gene therapy. So here we go.
Tom is 35.
Hello.
Overall, he's a pretty good guy. He helps his sick mother. He often cooks for his wife, Mary.
Huh?
He doesn't steal all the blankets, and he hardly ever snores. However, sometimes Tom has bad dreams. Recently, it seems Tom is making a mess in the kitchen, but Mary doesn't mind. Even when Tom encounters some difficulties in the bedroom, Mary still loves him just the same. Perhaps it's just a sign of aging. Over the past few years, Tom has been feeling rather down, and those shoulder aches might be related to a sports injury, but since Tom doesn't play sports, he decides to go for an orthopedic checkup. But the orthopedist doesn't see anything particularly wrong, so he refers Tom to a physiotherapist, who then refers him to a neurologist. Tom tells her about all the difficulties he's been having over the past few years, and how difficult it has been for him to take care of his mother. "Why are you taking care of your mother?" she asks. "She has Parkinson's disease," he replies. Now the neurologist understands. The nightmares, the loss of smell, the impotency and depression. Tom has been showing early signs of Parkinson's for 20 years. Parkinson's is genetic among Ashkenazi people. More than 1/3 of Ashkenazi Parkinson patients carry mutation in either the LRRK2 or the GBA genes. Moreover, the disease can be detected decades before the known motoric symptoms appear. Inside our brain cells, we find the important alpha-synuclein protein. When that protein is misfolded, it becomes toxic to the cell.
Okay, so we're going to hear a lot more about that toxicity in a few minutes, so I'll spare you that last minute. I do want to acknowledge all the really incredible people in my group who have done the work and much more that I didn't get a chance to tell you about today, and acknowledge the very generous funding that we have from foundations, from grants, and from private donations. And to thank all of you, and I'm looking forward to hearing the next.
Thanks very much, Karen. And that was quite compelling and hopeful data on the potential use of gene therapy to ameliorate hearing loss. And you did tee up nicely the next speaker. It's my pleasure now to introduce Dr. Mouradian. Dr. Mouradian is the William Dow Lovett Professor of Neurology at the Robert Wood Johnson Medical School and the founding director of the Robert Wood Johnson Medical School Institute for Neurological Therapeutics and Vice Chancellor for Faculty Development at Rutgers Biomedical and Health Sciences, Rutgers University. As an internationally recognized physician scientist, Dr. Mouradian's career has been focused on elucidating the molecular pathogenesis of Parkinson's disease and its treatment and complications, and to develop therapies both for disease modification and improved system control. Her seminal contributions have provided the rationale for the development of continuous therapeutic modalities for Parkinson's. To date, she's authored over 200 scholarly publications, edited 2 books on Parkinson's disease, and holds several patents. Her research is funded by multiple grants from the National Institutes of Health, the Michael J. Fox Foundation for Parkinson's Research, the American Parkinson's Disease Association, and other funding modalities. It's my pleasure now to introduce Dr. Mouradian. Maral.
Thank you, Vince, and thank you for the kind introduction. I also would like to thank you for inviting me to speak at this symposium series. I would also like to invite the organizers at Tel Aviv University for the collaboration with Rutgers to develop this symposium series. I'm delighted to be here. This has been a wonderful opportunity for me to get to know Karen. We actually had a nice chat about 10 days ago, and we worked together to plan our presentations. So today I will be speaking about gene therapy for treatment complications in Parkinson's disease. So to help explain what I mean by treatment complications, I will begin with a brief background about the disease. And I must say, Karen's introduction, the cartoon, was wonderful to give things in perspective. So Parkinson's is actually a common problem. It's the second most common neurodegenerative disease after Alzheimer's. We don't count them in billions as hearing loss is, but we're in the millions. And the prevalence is increasing. In fact, the numbers are expected to double in the next 30 years. 1% of the population over the age of 55 are estimated to have Parkinson's. It's a major cause of disability, as well as economic and societal cost. And it affects people from all walks of life. As you can see, these famous faces, all these individuals have or had Parkinson's disease. The primary pathology responsible for the classic motor symptoms, such as tremors, slow movements, and a shuffling gait, is in the base of the brain called the substantia nigra, where dopamine neurons are. And these neurons project to the striatum, which consists of the caudate and the putamen. That's where dopamine is released. And because of the degeneration in the base of the brain, there is depletion of dopamine in the striatum. If you take a cross-section of the brainstem here, this is what you see. Compared to a normal brain showing this nice, dark, melanin-containing dopamine neurons, you can see that in a Parkinsonian brain, there is massive depletion of these dopaminergic neurons. So if you can imagine the dopamine synapse, shown here, the yellow is the dopamine terminals where dopamine is synthesized and released to act on its postsynaptic receptors. In Parkinson's, there is massive depletion of these terminals, and therefore massive depletion of dopamine in the striatum in the order of 80% by the time the motor symptoms emerge. So the gold standard to treat the symptoms of Parkinson's is therefore L-DOPA, which is the precursor of dopamine, and has been the gold standard even after more than 50 years of discovering it. And it's a very powerful drug. And here is an example of what it can do. Here is a man with Parkinson's having difficulty walking with small steps. He has what we call freezing of gait. He's stuck in place, eventually gets going. And here is after he gets L-DOPA. You can see the stark difference. He can now swing his arms and walk independently. The problem with L-DOPA is after taking it for some time, this is what can happen. These are involuntary movements in this man affecting his entire body, unable to sit still. And in this man, affecting primarily his neck. So this is a treatment that helped this individual for a number of years, but is now left with these unacceptable involuntary movements. So this is what we call L-DOPA-induced dyskinesia, abnormal movements. It's a common problem. It occurs in about 50% of patients after 5 years of treatment and upwards of 90% by 10 years. It's a significant source of disability. And currently, the only medication that we, in fact, were the first to demonstrate over 20 years ago to have some anti-LID effect. It's not a full effect. And also, the medication has a number of side effects. And as a result, not very many patients can take it, particularly the older individuals. And a number of clinical trials trying to address a number of other transmitter systems have not yielded yet beneficial effect in suppressing these involuntary movements. And this condition is a common indication for brain surgery called deep brain stimulation. Now, for L-DOPA-induced dyskinesias to develop, you need two things. One, severe denervation, in other words, loss of dopamine neurons. And two, chronic repeated L-DOPA administration. So the combination of these two events results in L-DOPA-induced dyskinesia. The focus of this investigation that I'll be talking about is a chronic transcription factor called delta phosphate. And the reason for that, we and others have found that when there is denervation, delta phosphate expression goes up in the striatum as seen with these brown spots in immunohistochemistry. What is even more interesting is that when denervation is combined with repeated L-DOPA administration, delta phosphate expression goes up even much higher. So the question is, does this have anything to do with the development of the involuntary movements? In other words, can delta phosphate be the driver or the originator of involuntary movements? To answer this question, we tested in 2 animal models of Parkinson's. First in rats by injecting the toxin 6-hydroxydopamine in the medial forebrain bundle, which connects the dopamine neurons in the nigra to its destination in the striatum. As you can see here, the injected side, we do it unilaterally, you see massive loss of the dopamine neurons compared to the intact side where you see these dopaminergic neurons that are stained with tyrosine hydroxylase. So using this model, we tested whether overexpressing delta phosphate using an adeno-associated virus that Karen described to see if we can induce involuntary movements in the rats. Here is the experimental paradigm. First, we inject 6-hydroxydopamine unilaterally in the brain, in the striatum. We wait 3 weeks for the lesion to take hold. Then we test the animals with a subthreshold dose of apomorphine, which is a dopamine agonist. And what that does, it causes the animals to rotate because it's a unilateral lesion. And this is important to demonstrate that the lesion actually took place. And in our hands, this translates to upwards of 95% of dopamine neuron loss. Then we wait another week, and then we give 1 dose of L-DOPA as a test to make sure at baseline the animals do not exhibit involuntary movements. And then we divide them into 2 groups. The blue group receive chronic L-DOPA treatment. By that, I mean twice daily injections for 4 weeks or saline. And the other group were injected with an AAV delta phosphate expressing vector in the striatum on the lesion side, or the control vector just expressing GFP. Wait 4 weeks for the transgene to express. And then we test the animals just once weekly with L-DOPA to see if they would exhibit involuntary movements. And then at 12 weeks, we sacrifice and we look at the brain. So the difference between the blue group is that they're repeatedly receiving L-DOPA twice a day, and the red group is not. And here is what happens to their behavior. Focus on the far right, the blue curve first. Those are the animals that received chronic L-DOPA. As we expect with the blue curve, there is increased demonstration of involuntary movements on a number of measures that we use. We have a scale for involuntary movements in the rats called AIMS, their limbs, their mouth, as well as torsion of their body. So this is known, this is expected because they are denervated and they received repeated L-DOPA administration. What is more interesting is the red curve. These are the animals that received AAV delta phosphate in the striatum without chronic L-DOPA administration, exhibiting involuntary movements, just like L-DOPA does on all the measures, compared to the control vector injected animals in black. So this shows that the mere injection or overexpression of an AAV expressing delta phosphate in and of itself can induce involuntary movements in the rat. We wanted to make sure that there is indeed expression in the brain from these vectors. This is a delta phosphate stain in the AAV delta phosphate injected brain compared to the control brain. And we also confirmed this by Western blotting, delta phosphate injected ipsilateral brain versus the contralateral brain versus the control virus injected. So the question is, does the same phenomenon occur in the non-human primates? Obviously, the brain there is much more closer to the organization and the circuitry in the human brain. So it was important for us to demonstrate this in a non-human primate model of Parkinson's. And in this case, we administer another toxin called MPTP, which also kills dopamine neurons. And we administer it systemically. So the entire brain, both sides of the brain are exposed to the toxin, and both sides of the nigra are lesioned. So after repeated administration chronically for some time until the animal develops stable, significant degree of Parkinsonism with no L-DOPA treatment, that's important, then we just tested once weekly to see if with L-DOPA to see if they have involuntary movements at baseline as a control. And then the animals were subjected to implantation of a chamber device because some of them also had electrophysiological studies. Wait 4 weeks to recover. Then through the chamber device, they received AAV delta phosphate or AAV GFP virus injection bilaterally in this case, because the disease is bilateral in this model. Wait another 4 weeks for the transgene to express, and then test weekly the response to L-DOPA injections. This is a diagram where the virus was injected throughout the striatum of the animal, both in the caudate and in the putamen. Here is their behavior. The red curve are the animals that were injected with AAV delta phosphate, and you can see that they exhibit significant degree of L-DOPA induced dyskinesia, either the total L-DOPA, the LID score, or the peak LID score. And this is just a time course of LID exhibited with successive weeks of testing. What is equally important here is that despite the ability of delta phosphate to induce these dyskinesias, it had no effect on the Parkinsonian signs of the animal, neither at baseline without L-DOPA, nor after L-DOPA. This is important because clinically, there is a sort of a dichotomy between Parkinson's signs and the dyskinesias. By that I mean, when we try to treat Parkinson's signs, if the patient is prone to developing dyskinesia, dyskinesias can get worse. On the other hand, when we try to reduce the dyskinesias by decreasing, let's say, the L-DOPA dose, the Parkinson's symptoms can get worse. So it's important for us to find a treatment that can impact dyskinesias without affecting the anti-Parkinson effect of L-DOPA. And this will become more obvious in the next segment. So again, in these primates, we wanted to make sure that there is indeed expression of delta phosphate in the striatum that was injected compared to the control vector injected animals. This transgene delta phosphate is expressed in neurons co-localizing with the marker NUN. There is massive increase in the number of delta phosphate positive neurons, and this is also confirmed by Western blots. Again, these are delta phosphate injected animals compared to the control vector. As expected, there is significant increase in delta phosphate and also changes in a number of signaling molecules downstream from delta phosphate, including phosphodop32, CDK5, and forceful ERK. And there is also correlation between the degree of delta phosphate expression and the severity of L-DOPA induced dyskinesias, as well as with some of the other markers. So far we established that the mere overexpression of delta phosphate in the absence of repeated chronic L-DOPA administration can induce involuntary movements, both in the rat and the non-human primate. So the next step, the logical step, is to use this information and see if we can prevent L-DOPA-induced dyskinesia by inhibiting delta phosphate expression. So to do that, we designed an AAV vector expressing shRNA against delta phosphate. Now, delta phosphate is a splice variant of phosphate. It's missing the 101 C-terminal amino acids because of a splicing change that results in a premature termination codon. And this is the splice junction. So for us to target delta phosphate only, we generated a vector that targets this particular splice variant only. And here is the sequences. We initially tested 3 different sequences in a cell line and found that this particular sequence, siRNA number 1, is the most effective in a cell line. And then put it in this vector, AAV vector, versus the non-targeting control vector. And we use this in the rat model in this case. And here is the timeline of the studies. So we lesion with the 6-hydroxydopamine. 5 days later, they were injected with this AAV shRNA, either delta phosphate or the non-targeting vector. Waited 3 weeks, did that apomorphine rotation test, waited another week, and then started testing the response of these animals to L-DOPA. Here is what we found. So compared to animals that were injected with non-targeting control vectors, given L-DOPA, they exhibit involuntary movements as expected, whereas inhibiting delta phosphate with shRNA significantly repressed the degree of involuntary movements in these rats. Again, importantly, the anti-Parkinson effect of L-DOPA, or the baseline Parkinson signs of the animal, what we call hypokinesia tests, was not affected by inhibiting delta phosphate. Again, this is critical when it comes to clinical translation, because there are ways of inhibiting dyskinesias. For example, one theoretically can give a dopamine antagonist to inhibit dyskinesias, but what happens in that case, the Parkinson's symptoms get much worse. So that's an unacceptable strategy. We need to have a way to inhibit dyskinesias and keeping the anti-Parkinson effect of L-DOPA intact. And this strategy accomplishes that. We looked at the brains, and by Western blots, as expected, the delta phosphate shRNA injected animals had significant decrease in delta phosphate, as well as changes in the downstream signaling molecules. These are the quantifications, and these are the correlations with abnormal involuntary movement scales. Also, importantly, there was no tissue damage to this manipulation. The number of neurons in the striatum, either the delta phosphate or the non-targeting vector, there were no differences. And also there was no neuroinflammation, either by GFAP looking for astrocytes or by IBA1 looking for microglia. So the tissue was intact. There was no ill effect either from the AAV or to delta phosphate downregulation. So in summary, we've shown that the increase in striatal delta phosphate in and of itself plays a major role in the development of L-DOPA induced dyskinesia. And the flip side of that, knocking down delta phosphate by gene therapy can mitigate these involuntary movements. And therefore, this provides the rationale for reducing striatal delta phosphate in Parkinson's patients with troublesome dyskinesia. I would like to close by acknowledging my team members, and in particular, Ji Zhang, Zhongmin Wu, and Su Park, who were closely involved in this particular project. My close collaborator, Stella Papa, who's a neuroscientist and a primatologist at Emory and the Yerkes National Primate Research Center. And my other collaborator on this project, Hideki Mochizuki at the University of Osaka, and my funders, including the NIH, Michael J. Fox Foundation, the American Parkinson's Disease Association, and other foundations. Thank you very much.
And thank you, Dr. Mouradian. That was a final review of hopeful gene therapy approach for Parkinson's. I have a number of questions to ask, and I want to remind audience members, if you do have a question, please go to the bottom of the screen on the Q&A section, and there you'll touch that button and you can ask a question. First question is directed towards Dr. Mouradian and more of a mechanistic question. The question is, what's known about how delta phosphate causes LID? Is the delta form of the protein expressed along with the normal native full-length protein?
Well, it's primarily the delta phosphate, the variant that is massively upregulated. What causes it? We think it's all those downstream signaling molecules that I went through very, very quickly, and other transmitters that are regulated. It's pretty complex. I didn't go into those details. There is the direct pathway and the indirect pathway and so on going out of the striatum. So it's probably not just one target or one molecule, but the delta phosphate itself seems to be the initiator of these downstream signaling cascades. And so that's why I think by reducing its expression, you're modulating the downstream pathways and causing the mitigation of the involuntary movements.
And then just a follow-up, I guess, is the unilateral delta phosphate AAV treatment sufficient to reduce dyskinesia on both sides, or is it?
Well, the unilateral was in the rat model. So in a human being, most of the time they will have bilateral symptoms, particularly when they have dyskinesia. So bilateral treatment will be needed.
Thank you. The next question's directed towards Karen. Karen, two sort of related questions. First, are any of the genetic causes of deafness, hearing loss reversible? And with respect to the delivery of a gene therapy, must it be initiated prior to profound hearing loss to be effective?
Thanks, Vince. And first, I just want to say, Dr. Mouradian, that was excellent. Really enjoyed hearing that work that you're doing. And I looked at the papers and really exciting. So, well, unfortunately, the genetic causes are not reversible. There is preventable. There's, albeit rare in most places in the world, but mitochondrial mutations that lead to hereditary hearing loss. And if those can be discovered in advance, you can avoid, and the hearing loss is caused or triggered by giving aminoglycosidases. Now, those are antibiotics that are not necessarily given in many parts of the world, but places that they are, if you can avoid giving them, and you know that there's mitochondrial mutations. So that's a preventable form. But other than that, all of the other genetic forms, and the problem really is because those hair cells that I showed you earlier, we're born with about 16,000 of those in each one of our ears. And once they die, they are post-mitotic, so they don't regenerate. In fact, one of the major areas of research that are ongoing in our field, and I didn't get into that, is trying to regenerate the hair cells. And I already know, and in the next question, if the gene therapy would have to be initiated prior to a profound hearing loss. So the answer is yes, because the problem with most of the causes, or at least the end result of hearing loss, is that you lose your inner and outer hair cells. And those are the cells that are essential for us to hear. And once they die, there's no way to resurrect or regenerate those. And that's why there's a hope to be able to do that kind of, to find the key factors that could promote regeneration. Now, you could imagine a situation that even if the damage has already been done, if you know what those factors in, and we have ways to introduce gene therapy, that you could actually introduce those factors in and you can do the reverse. But we're not there yet, unfortunately. So that becomes an issue, particularly in cases where children who are born deaf, in the case of congenital hearing loss, because it's hard to imagine that we would go in utero. And so that's why I talked about doing newborn hearing screening testing that's done at the hospitals and fusing it to being able to determine very early on what's the cause of hearing loss. And that can really help in terms of the type of therapy that these children would be getting early on.
Thanks. I'm going to tack on a question to a question I see here. This is also for Karen. Karen, what kind of ethical issues have any of you faced in your research? And just to tack on a little bit to that, I understand that in the deaf community, there's sort of a culture of not wanting interventions, and certainly these physical modalities for improving hearing in a subpopulation of the deaf community. So answer the question, are there ethical issues you've confronted? And second, does the approach of using gene therapy early in life, is that maybe a more acceptable approach for those who have concerns?
Well, I want to emphasize, first of all, we have enormous respect for whatever point of view that you have. And that population that you talk about is small. It tends to be families where the hearing loss is inherited. And in fact, early on, there was a lot of resistance to cochlear implants, but many of them have come around, and there is an effort to use cochlear implants. A lot of it has to do with the fact that you don't want to alienate those that are in your community if they don't, if they're not using the hearing aids. And you could also, you almost can become like two separate worlds, and they want to make sure that their community stays integrated. But unfortunately, the much larger part of the world of people who have a hearing loss are those who are not in that group. So from their perspective, the ethical issues are more in any standard ethical issues that we might have with respect to gene therapy, some of the fears that people might have, how long it's going to last. But all of the work that's being done, of course, in our lab, in Dr. Mouradian's lab, and all the other labs that are doing this are taking very good care of going through all the ethical guidelines and regulations that need to be done. We work very closely with the institutional review boards in the Helsinkis when it comes to working with animals, having the proper IACUCs in place. So there haven't been any problems from that perspective at all.
I think that's true at Rutgers as well. Back to Dr. Mouradian, what is the current thinking or outlook on glucocerebrosidase delivery as a possible therapeutic or prophylactic for Parkinson's disease?
That is an interesting question. As Karen mentioned in her presentation, GBA mutations are a well-known cause of Parkinson's. So the approaches that are being tested there are actually using other modalities to enhance the function of glucocerebrosidase. It's a complex system there because it also relates to synuclein pathology, which obviously I did not talk about our number of projects that we do related to synuclein, and also lysosomal function. Because with GBA mutations, there is a significant lysosomal dysfunction. So there are a number of ways that the GBA mutations can be addressed, in addition to gene therapy. Obviously, it's easier to develop small molecules than gene therapy, and you can see the pains that Karen and I have to go through to develop a gene therapy. It has been a long and windy road, but we're making significant progress. So if there is a way to enhance GBA activity in those individuals, and there are a number of companies doing that actually, that will be easier. But in parallel, there are also efforts using gene therapy as well.
Just going back to delta phosphate for a second, I think I read that overexpression of delta phosphate is also connected with addiction, severe addiction. And do you think this approach could help with severe addiction as well?
Very good question. Yes, quite likely. Yeah, it does go up in a number of other manipulations. Drugs can do that. So yes, that is an interesting possibility.
Right. I guess I have one final question for both of you, and that is not speaking to the specific gene sequence they're using for therapy, but the various modalities to deliver genes have improved greatly over the last couple of decades. And do you see that as, sort of helping to create a golden opportunity in terms of gene therapy writ large? And if so, are there even better gene delivery modalities coming on the scene in the coming years? So I'll start with Karen and then Maral.
So I want to add that there are many already about 50 or so companies that have really entered into the space in the past couple of years. So I think that there's a lot of promise. I think one of the limitations that we might see with respect to gene therapy for specific genetic mutations is that it might be only like for many rare diseases that we're talking about, it might be just a handful. So for example, it doesn't seem that practical that a company who is starting out is going to develop already something for SYN4 when there's 5 or 6 families worldwide. It might be different if we're looking at genes like myosin 6 or TMC1, that there might be tens of thousands in the world. And so that's part of an issue that's certainly going to come up. And I think as we get better at doing the gene therapy, then there might be ways to be able to have more. In each case, it'll be a specific gene, but because the details of the technological breakthroughs will have been worked out so well that it'll be much more routine and standard to each time introduce a new gene. We're certainly not there yet. The other issue is, like what Dr. Mouradian spoke about, there are small molecules that are being tested. And so there's a number of clinical trials that are going on there. So I think that the bottom line will be for hereditary hearing loss or environmental hearing loss, that it's not going to be one size fits all. There will be a precision medicine approach. Some will do better with AAV clinical trials that will become relevant and will move into the clinic. Others, it'll be small molecules. Others, it might be regeneration that needs to be done. It'll very much depend on the age of the onset, what the progression of the hearing loss is. So it gives us a lot of challenges and a lot of work for the future.
Okay. By the way, you just managed to answer a question that popped in while you were speaking.
Okay.
There are, in fact, small molecules out there that approaches towards treating deafness. Dr. Mouradian, do you want to say a few words about different modalities to deliver genes?
So what determines the modality of treatment or delivery is what the disease is, where it affects, what the natural history is. So in the case of the condition that I just described, L-DOPA induced dyskinesia, which appears to originate from the striatum, so that's where you target. And there are ways of doing it in human beings, and it is being done. And the way to do that is what's called convection enhanced delivery. And you can even do that under live MRI guidance. So you know exactly where what you're infusing is going and covering most of the brain region that you want to cover. So it's doable. So it's very localized. But for a genetic disease, for example, that is systemic, then you need to have a different strategy. Or a brain disease, for example, in SMA or other conditions, then you may have to think about intrathecal delivery of the transgene. So it really depends on what your target is, and design the delivery accordingly, and how often you need to give it, and how much. All these need to be worked out.
I'm intrigued by this notion of MRI imaging and gene therapy. It's a double targeting of a therapy. I like that. Well, I want to thank both of you for both your excellent presentations and your thoughtful answers to the questions from the audience. This will conclude our session on gene therapy. I did want to point out that this session has been recorded. This session and the prior 4 sessions can be found on a website, I think both at TAU and at Rutgers, but I'll give you the Rutgers site. It's go.rutgers.edu/Rutgers-TAU, and we can provide this information to the audience. Our next seminar series will be on May 11th, so look out for that next presentation. It will be hosted on the TAU side. And I want to thank you all for your attention and thoughtful questions. Have a good day.
Thank you. Great to be back in New Jersey again.
Yeah. Great.
Bye, Karen.
Bye, everyone.